Welcome to another lecture by MedicoMedics, Genetics Chapter 9, Genomics. In this lecture, we will discuss what is genomics, talk about the Human Genome Project, mention some applications, some key tools, some types of genomic studies, challenges, the future, and end with a summary. Now, genomics is the study of an organism's entire genome, including all its genes and non-coding regions. Focus areas include genome sequencing, so reading the entire genetic code; comparative genomics, comparing genomes across species; and functional genomics, understanding gene expression and interactions. What are some applications of this? Well, we use it in personalized medicine, disease research, and evolutionary studies. So genomics is like studying an entire library of DNA to understand how it works, evolves, and influences health. Now, the Human Genome Project was a global effort to sequence all 3 billion base pairs of human DNA, and it started in 1990 and completed in 2003. Key achievements: it identified approximately 20 to 25,000 human genes, and it mapped all genetic variations. The impact? Well, it laid a foundation for personalized medicine and enabled genome-wide association studies. So it was like creating a detailed map of all human genes to better understand health and disease. High-throughput sequencing, so the next generation of sequencing, is an advanced technology to sequence DNA rapidly and at a large scale. So how does it work? Well, DNA is fragmented. Fragments are amplified and then sequenced in parallel. And then we use computational tools to assemble the sequence. The advantages: it's faster, cheaper, and more accurate than earlier methods, like, for example, the Sanger sequencing. So sequencing an entire genome now takes days instead of actual years. Some applications in medicine: identifying disease-causing genes like BRCA mutations, developing targeted therapies; agriculture, breeding disease-resistant crops, improving livestock genetics; evolutionary biology, so tracing human ancestry and migration patterns, studying genome evolution across species. Some key tools used in genomics: we have polymerase chain reaction, PCR, where we amplify specific DNA sequences for analysis; microarrays, where we analyze gene expression or detect genetic variants; CRISPR technology, so genome editing tool to modify genes with precision; bioinformatics, software and algorithms to analyze large genomic data sets. CRISPR has been used to treat genetic conditions, for example, like sickle cell anemia. Some different types of genomic studies. So we have whole genome sequencing, which sequences the entire genome to identify all genetic variations. Exome sequences focuses on protein coding regions of the genome. Genome-wide association studies identifies genetic variants associated with diseases by comparing genomes of affected and unaffected individuals. Examples: genome-wide association studies studies the identified genes linked to diabetes, heart disease, and Alzheimer's. Genetic variation and its implications. So we have different types of variations. We have, for example, single nucleotide polymorphisms, where we talk about single base changes. We have copy number variations, so duplications or deletions of DNA segments. We have structural variations, so larger changes in the DNA structure. Why is this clinically relevant? Well, for example, single nucleotide polymorphisms are linked to drug responses, something called pharmacogenomics, which we will talk about in an upcoming lecture. And we have variations causing genetic disorders. An example of this is single nucleotide polymorphisms in the CYP2C19 gene affect how individuals respond to the blood thinner clopidogrel. This is directly relevant, clinically relevant. Some challenges in genomics. So data volume. So sequencing generates massive data sets requiring advanced computational tools. Ethical concerns. Privacy of genetic information. Discrimination based on genetic data. Clinical translation. So bridging the gap between research and practical application in medicine. These are some of the challenges lying ahead. Now, what does the future hold? Well, some trends are personalized medicine based on individual genomes, advances in artificial intelligence and machine learning for genomic analysis, synthetic genomics, so designing and building artificial genomes. Potentials, well, it gives us the ability to maybe eradicate genetic diseases, revolutionizing drug development, and creating bioengineered organisms for specific purposes. This might be a bit scary, but, let's see. An example, AI tools are being developed to predict cancer risk based on genomic data. So, in summary then, genomics studies the entire genome to understand its structure, function, and evolution. High-throughput sequencing technologies have revolutionized how quickly and accurately we can sequence DNA. Applications span medicine, agriculture, and evolutionary biology. Challenges include data management, ethical concerns, and translating research into practice. So genomics is about studying all our DNA, using fast and advanced tools to improve health, food, and our understanding of life itself. That's the end of Chapter 9. Continue now to Chapter 10. Thank you for listening.